What is actually inside this cannabinoid oil? When people look at a CBD oil, they often see just one number on the label. A percentage. But the cannabis plant is much more complex than that. It does not contain only CBD. It contains a wide range of different cannabinoids, each with its own chemical structure, and researchers are still trying to understand what many of them actually do. In this episode, we are going to take a closer look at a laboratory analysis of the cannabinoid extract used in this oil. The purpose is not to claim that the oil can cure or treat specific diseases. The purpose is simply to understand what is actually present in the extract, and why some of these cannabinoids are interesting from a scientific point of view. And this particular analysis is quite unusual. CBD is, of course, the dominant cannabinoid. But after CBD, we find a remarkably high level of CBDV, followed by THCV. Those two cannabinoids are what really make this profile stand out. So let us begin with the numbers. The analysis shows CBD, 64.5%. CBDV, 9.79%. THCV, 2.36%. CBG, 1.133%. CBDA, 0.957%. CBE, 0.64%. CBV, 0.258%. And delta 9 THC, 0.138%. Together, the cannabinoids that the laboratory was able to quantify make up approximately 79.8% of the extract by weight. A number of other cannabinoids were also tested, including CBN, CBC, CBT, THCA, CBGA and delta 8 THC, but these were below the laboratory's limit of quantification. So now, let us look at the most important cannabinoids one by one. CBD, 64.5%. We begin with CBD, or cannabidiol. CBD is by far the most extensively studied cannabinoid in this analysis. The strongest clinical evidence is found in the field of neurology. Pharmaceutical grade, precisely dosed CBD is already used as an approved medicine for certain severe forms of epilepsy. That tells us something important. CBD is not only something discussed in alternative health circles. It is also a substance being taken very seriously in modern medical research. And the research does not stop with epilepsy. CBD is being studied in relation to anxiety, stress, inflammation, sleep, pain, neurological conditions and several psychiatric disorders. Anxiety and mental tension are particularly interesting areas. There have been controlled human studies with encouraging findings, although the research still does not give us one simple answer about who may benefit, at what dose, and under which circumstances. When we look at user experiences with CBD, some of the same themes appear again and again. People often describe using CBD in connection with stress, restlessness, sleep difficulties, and the desire to relax. Personal experiences are not the same as clinical evidence. But they can still be useful because they help researchers identify areas that may deserve further study. CBDV, 9.79%. Now we come to one of the most interesting parts of this analysis. CBDV. Cannabidivarin. This extract contains 9.79% CBDV. That is a substantial amount. CBDV is chemically related to CBD, but it is not the same molecule. It belongs to a group known as the varin cannabinoids. CBDV has attracted particular scientific interest in research involving the brain and nervous system. Researchers have investigated CBDV in areas such as epilepsy and autism. In studies involving people with autism spectrum disorder, researchers have found measurable changes in brain systems associated with glutamate and GABA. These are two important signaling systems in the brain. Very simply put, one is heavily involved in excitatory activity, while the other is involved in inhibitory activity. The balance between these systems is therefore very important in neurological research. Brain imaging studies have also suggested that CBDV can influence communication between certain regions of the brain. That does not mean that CBDV has been proven to treat autism. But it does show that CBDV can produce measurable biological effects in humans. CBDV has also been studied in epilepsy. In one larger clinical study, seizure frequency fell among participants receiving CBDV. However, seizure frequency also fell by almost the same amount in the placebo group. So researchers could not conclude that CBDV was superior to placebo in that particular trial. And that is an important part of science. Some findings look promising. Other studies show us that the picture is more complicated. But regardless, a CBDV concentration of almost 10% makes this cannabinoid profile quite unusual. THCV, 2.36%. The next cannabinoid is THCV. Tetrahydrocannabivarin. THCV sounds very similar to THC, but the two substances have different pharmacological characteristics. THCV has received particular attention in research involving metabolism, blood sugar regulation, insulin, appetite, and body weight. There has even been a randomized study involving people with type 2 diabetes. Researchers studying THCV observed interesting changes in fasting blood glucose and certain markers related to pancreatic function. That does not mean that THCV is a treatment for diabetes. But it is important because we are talking about controlled human research, not only laboratory experiments. More recent studies have also examined combinations of CBD and THCV in relation to body weight, waist measurements, and metabolic markers. The field is still young. But it demonstrates just how differently individual cannabinoids can behave. When people think about cannabis, many immediately think about increased appetite. Yet THCV is being investigated for biological mechanisms that may in some situations point in the opposite direction. In user reports, THCV is also often described as a more clear-headed and daytime-oriented cannabinoid. Some people report feelings of focus, energy, and mental clarity rather than heaviness or sedation. CBDV is sometimes described in a similar way. That is one reason why the combination of nearly 10% CBDV and 2.36% THCV makes this extract particularly interesting. CBG, 1.133%. Next comes CBG. Cannabigerol. CBG is sometimes described as a kind of parent cannabinoid because compounds related to CBG are used by the cannabis plant as biochemical starting points for several other cannabinoids. CBG is being investigated in relation to inflammation, pain, gut function, the nervous system, stress, and anxiety. Human research is also beginning to appear. In one small double-blind study, participants were given 20 milligrams of CBG. Researchers reported reductions in self-reported anxiety and stress compared with placebo. It was a small study, so the result should not be exaggerated. But it is still interesting because research into minor cannabinoids has historically relied heavily on cell and animal studies. We are now beginning to see more research involving actual human participants. In this extract, CBG is present at just over 1%. That is much less than the CBD or CBDV level, but it is still a measurable part of the cannabinoid profile. CBDA, 0.957%. The next compound is CBDA. Cannabidiolic acid. CBDA is a natural precursor to CBD. When the cannabis plant is heated, CBDA can be converted into CBD through a process called decarboxylation. But CBDA also has biological properties of its own. One of the most interesting areas of research involves nausea and vomiting. Laboratory and animal studies have suggested effects involving the serotonin system, particularly a receptor known as 5-HT1A. This has created scientific interest in CBDA in relation to nausea, including nausea associated with chemotherapy. But it is important to understand that much of this research is still preclinical. Results observed in animals cannot automatically be assumed to occur in humans. Still, with almost 1% CBDA, this cannabinoid is clearly present in the extract. CBE and CBV. The analysis also shows 0.64% CBE and 0.258% CBV. Here we enter the world of some of the much less studied cannabinoids. CBE stands for cannabinoid. CBV stands for cannabivarin. At the moment, there simply is not enough human research to make strong claims about exactly what these cannabinoids do. And that is actually an important part of the story. The cannabis plant contains many compounds that science still understands only partially. That does not automatically mean that these compounds have extraordinary properties. It simply means that the research is not finished. THC, 0.138% in the extract. The laboratory also measured delta-9 THC at 0.138% in the analyzed extract. That is a very small proportion compared with CBD at 64.5%. And it is important to remember that this analysis was performed on the concentrated extract used as a raw ingredient. If that extract is later diluted into an oil, The concentration in the finished product depends on how much extract is used in the final formulation. What does the overall profile tell us? Now let us step back and look at the entire picture. This is not simply an extract containing 64.5% CBD. It also contains 9.79% CBDV and 2.36% THCV. Together, those two varin cannabinoids make up slightly more than 12% of the extract. That makes this profile very different from a product based on pure CBD isolate. And this raises an interesting question. Can different cannabinoids influence one another? This is where we often hear the term entourage effect. The idea is that several compounds from the cannabis plant may interact and create biological effects that differ from those of a single isolated compound. It is an interesting theory. But it is also sometimes promoted more strongly than the evidence currently supports. We do not yet know whether a broad cannabinoid profile is generally better than isolated cannabinoids. The outcome may depend on the ratio between the compounds, the dose, the individual person and many other factors. So the most accurate way to describe it is that cannabinoid interactions remain an important area of research. Which areas are most interesting for this profile? If we look at existing research, while keeping user experiences as a separate category of information, several areas stand out. The first is stress, anxiety, and mental restlessness. CBD is the main cannabinoid of interest here, while CBG adds another research dimension. The next area is mental clarity, focus and daytime function. This is where CBDV and THCV make the profile particularly interesting. That does not mean that these cannabinoids should be described as treatments for conditions such as ADHD. There is not enough evidence for that. But both the pharmacology and user experiences surrounding CBDV and THCV make focus and mental clarity interesting areas for future research. The third area is neurology. Here we have CBD, with its strongest clinical evidence in specific forms of epilepsy, together with CBDV, which is being studied in epilepsy, autism, and brain signaling. The fourth area is metabolism. THCV is being investigated in relation to blood glucose, insulin, body weight, and metabolic disorders. The fifth area is inflammation and pain. CBD and CBG interact with biological systems involved in both inflammation and pain perception. Laboratory and animal research is extensive, while results from clinical studies in humans remain much more mixed. What about sleep? Many people automatically associate CBD with sleep. But this particular cannabinoid profile is, in my view, not primarily a classic sleep-oriented profile. For example, CBN was not detected above the laboratory's quantification limit in this analysis. CBD is being studied in relation to sleep, but controlled studies have produced mixed results. For some people, any benefit may be indirect. If a person experiences less stress or mental restlessness, that may naturally make it easier to relax. But that is not the same as saying that CBD is a sleeping medication. What about mental health conditions? CBD is also being studied in psychiatry. There is research involving anxiety and psychosis. The findings regarding anxiety are interesting. For psychosis and schizophrenia, the picture is more mixed. Some studies have reported promising effects, while others have not demonstrated a clear clinical benefit. So this remains an interesting but unresolved field of research. And with serious mental health conditions, cannabinoids should not be used as a replacement for professional medical treatment without appropriate medical guidance. And what about cancer? Cancer is probably one of the areas where it is most important to separate different types of research. For many years, scientists have studied cannabinoids in cancer cells and animal models. In those settings, researchers have examined effects on mechanisms involving cell growth, programmed cell death, inflammation, and the blood supply to tumors. That is interesting science. But an effect on cancer cells in a laboratory dish is not the same thing as curing cancer in a human being. Clinical cannabinoid research is also taking place. But in people living with cancer, some of the more established areas of research focus on symptoms associated with the disease and its treatment. Examples include nausea, appetite, pain, sleep and quality of life. CBDA is especially interesting in this context because of the research involving nausea. CBD is also being studied in several clinical settings. So cannabinoids and cancer are absolutely a real field of scientific research. But it is important to distinguish research into cancer biology from evidence for an actual cancer treatment. Those are two very different things. The overall assessment. So if we summarize this entire analysis in just a few sentences, we are looking at a cannabinoid extract that is primarily dominated by CBD. But at the same time, it contains an unusually high level of CBDV and a clearly measurable amount of THCV. That makes the profile particularly interesting from a research perspective in areas such as stress and mental restlessness, neurology, brain function, mental clarity, metabolism, inflammation, and several areas where research is still at an early stage. Perhaps the most interesting thing about this analysis is therefore not simply the number 64.5% CBD. It is the combination. 64.5% CBD, 9.79% CBDV, and 2.36% THCV. Together with the smaller cannabinoids CBG, CBDA, CBE, and CBV. That gives us a much more complex picture of the cannabis plant than the single word CBD. And perhaps that is one of the most fascinating things about cannabinoid research. The more scientists investigate the plant, the more obvious it becomes how much there is still left to understand. Final note. Before we finish, there is one important clarification. The laboratory analysis discussed here was performed on CBD extract PHEC 654, which is used as a raw ingredient in the oil. The analysis documents the cannabinoid profile of the tested sample and batch. The cannabinoid concentrations in a finished oil depend on the amount of extract used in the final formulation. This discussion of scientific research, biological mechanisms, and user experiences is intended as general, alternative and research-oriented information about cannabinoids. Christian Maersk Registered & Co. APS does not sell the oil as a medicine or pharmaceutical product and does not claim that the product can diagnose, treat, cure, or prevent disease. Research findings involving individual cannabinoids cannot automatically be transferred to this particular oil or to any individual person. The laboratory analysis tells us which cannabinoids are present in the analyzed extract and in what concentrations. It documents the chemical composition. It does not document a medical or curative effect. And that is why this presentation should primarily be understood as an invitation to learn more about cannabinoids and the research that continues to develop around them.